Okay, so now we're going to get into the nitty-gritty of. Okay, so let's take a step back for a moment and get ourselves situated with the light reactions and remember their relationship to the Kelvin cycle. So remember, all of this is occurring in the chloroplast. And what we're going to talk about is the details of this part right here of photosynthesis. Now, this part involves the absorption of light and water coming in and being split and oxygen leaving. And then the process of producing the ATP and the NADPH that drives the Kelvin cycle. So remember, the Kelvin cycle depends on the light reactions. And really, the light reactions kind of depend on the Kelvin cycle still working because the Kelvin cycle is going to provide the oxidized NADP plus and the hydrolyzed ADP plus the inorganic phosphate, which is going to be used to make the ATP. What happens with the lights at the pigments? So now we're working into the detail actions. So if we look at this, the pigment, in this case, it's showing chlorophyll right here. It's going to be at a ground state. So that's opposed to an excited state in terms of the electron that's going to be passed. Now, the pigment molecule, remember, is going to absorb the light. We can see the photon being absorbed. Okay, and then there's going to be electrons that began at the ground state, but however, the light is going to create an excited state of the electrons. During this process, some heat is released or lost, right? And then, of course, the photons are going to eventually fluoresce. So this is called an afterglow. Now, not all photons actually do this, but the most important thing here is this chlorophyll molecule. We're going to be referring to a special set of chlorophyll molecules in a complex of proteins, which is going to be involved in the electron being converted from a ground state to an excited state and then passed off to something. Okay, so here this represents the energy of the electron. So the energy of the electron starts from low and then goes to high energy. So there's two types of photosystems in the thylakoid membrane of the chloroplast, and these are involved in the harvesting of the light and the passing of electrons. So the photosystem is basically a complex of proteins with pigments embedded in them. And all of this is embedded in the membrane of the. So this kind of shows what your book is depicting a photosystem as. So the photosystem represents this whole thing right here. And of course, this is the thylakoid membrane, very similar to the cellular membrane with the phospholipids and the tails, right? So here, the photosystem, the purple represents the proteins, and then the green represents the pigment molecules. And you can see the pigment molecules are absorbing the photons of light, which will be the energy from this is going to be transferred to a special pair of chlorophyll A molecules in the center of the photosystem. Now, these special pair of chlorophyll molecules are going to accept these electrons or photons from the light that was harvested from the other pigment molecules. Here, electrons are going to be excited. So they're going to be transferred from a ground state to an excited state and then passed off to a primary electron acceptor. Now, this primary electron acceptor may be different depending on the type of complex that we're dealing with. However, what matters here is that these represent the photosystems, and these are basically light harvesting complexes with a reaction center. Okay, and it's the reaction center where the electron becomes excited and then is passed on to the primary electron acceptor. Now, there are two types of photosystems, like I said. Both of them are made up of similar structures. So they're both made out of proteins and pigment molecules, and they both absorb light, and they both have a reaction center. However, there are some slight differences. Now, this kind of shows the structure of a photosystem. This shows the ribbon diagram, which we already kind of talked about when we talked about proteins. Again, it's a protein, and then you can see the chlorophyll molecules in green embedded in the thylakoid membrane. And you can see there is a thylakoid space and the stroma side of this complex. Now, let's talk about the photosystems and the flow of electrons. So there's two routes in which the electrons can flow. There's a linear route, and then there's a cyclic route. They both produce somewhat different things, and they're both made for slightly different purposes. So we're going to discuss those a little bit more in detail. And then, again, there are two types of photosystems. There's photosystem 2 and photosystem 1, or PS2 and PS1, also called P680 and P700. So the 680 and the 700 represent the optimum wavelength in which absorption occurs. Okay, so photosystem 680 absorbs best at 680 nanometers. Photosystem 1, which is P700, is going to absorb better at 700 nanometers. Now, PS1 and PS2 are both involved in linear electron transport. However, only photosystem 1, or PS1, is involved in cyclic electron flow. Now we're going to talk about linear electron flow first because it makes more sense. It's a little bit more complex because it involves both photosystems. But after we get through that, cyclic electron flow will seem a lot easier. All right, so we're going to start with photosystem 2. Now, photosystem 2 was discovered after photosystem 1. But in linear electron flow, it's photosystem 2 that really starts the process. So we're going to talk about that one first. PS2. So this represents the photosystem here. So you can see the reaction center with this pair of special chlorophyll molecules. And you can see electron being excited and then passed off to a primary electron acceptor. But the first step, of course, is the absorption of light. So basically, light is absorbed, these photons, and are going to be transferred to this pair of the chlorophylls in the center. And then the electron is going to be excited and then passed to the primary. During this process, water is also split. So that's where the electrons are coming from. Okay, so the electrons come from water once it's split. And here, once water is split, the hydrogens are going to be stored in the chloroplast and be used later. And then the oxygen is going to be released, as of course it's a gas, it's going to be released through the stomata of the plant leaf. What happens next is that we have this electron that is passed off to a primary electron acceptor. This PQ stands for plastoquinone, so that's basically going to transfer that electron to a cytochrome complex. And this is really the electron transport chain right here. So if you remember the electron transport chain from cellular respiration, you will know exactly what's happening here. So basically, the electrons are going to be passed from one complex to another in the electron transport chain. A hydrogen ion gradient is going to be built across the membrane, driving the synthesis of ATP by chemiosmosis. The proton motive force is also involved. And ATP synthase, it's just another type in plant. It does the same thing. It's going to combine inorganic phosphate with ADP to make ATP. Now we have plastose. That's going to be passing the electrons to the reaction center in PS1 or P700. P700, or this PS1, also absorbs some light and energy, and then an electron is going to be excited. Well, it's passed on from PS2. And then these electrons are going to become excited and transferred to the primary electron. But then the electron is going to be passed to ferredoxin, And then ferredoxin is going to transport the electrons to NADP plus reductase, which is an enzyme that reduces NADP plus to NADPH by adding the hydrogen. Okay, so this is all linear electron flow. It involves photosystem 2, photosystem 1, with the electron transport chain in between producing ATP. At the end of the process at photosystem 1, we have the production of NADPH. Okay, so during linear transport, ATP is produced, NADPH is produced, water is split, and oxygen is released. So this kind of shows that process. So we have the proton, it's moving from a lower energy to a higher energy against the force of gravity, if you will. And then the electrons will be passed slowly, remember, one by one. This is a stepwise fashion to control it, right? So again, it's going to break the fall of the electrons, and this produces the ATP, very similar to the ATP production in aerobic respiration in the mitochondrion. And then the electrons will be passed up again and then back down to NADP plus reductase to produce NADPH. So here, it's basically showing it as analogous to a production mill. Okay, now we're going to talk about cyclic electron. So cyclic electron flow only involves photosystem 1. So basically the electrons are going to be cycled from the electron transport chain from ferredoxin to the reaction center of the photosystem 1 via plastocyanin, or the PC that we mentioned before. So this process produces ATP, however, it does not produce NADPH, even though it involves photosystem 1, and we'll see why. Now here, no oxygen is released because, remember, oxygen is released during the splitting of water, which occurs at photosystem 2. So this kind of shows that process. So you can see, like, shaded is the photosystem 2, which is not involved in cyclic electron flow. So we can see the electron transport chain is going to produce ATP, okay? Plastocyanin is going to transfer the electrons to photosystem 1. Electrons are going to be excited and passed to the primary electron acceptor, which will then be passed to ferredoxin and then back through the electron transport chain. You can see that ferredoxin is going to bring the electrons back to the electron transport chain rather than to NADP plus reductase. So the process here is only producing ATP. So this is going to occur when the cell needs a little bit more ATP than it does NADPH. So if you can kind of think about it, there are other things that occur in the chloroplast, right? So it's not just photosynthesis that occurs in the chloroplast. Remember, there's DNA in the chloroplast. Proteins are going to be made in the chloroplast. There's energy that is going to be required to do other things. Okay, so again, the chloroplast is ultimately going to need more ATP. So this is the way to produce more ATP without producing excess NADPH, which may not be needed for the Calvin cycle. Okay, so that's cyclic. Now let's compare chemiosmosis in the chloroplast with the chemiosmosis in the mitochondrion. So remember, both of them involved in electron transport chain, right? And both of them involve the production of a chemical proton gradient and the proton motive force and ATP synthase. So this is where it's happening at least in the chloroplast. Okay, so remember, in the mitochondrion, it occurs at the membrane of the mitochondrion. And ATP is produced in the matrix of the mitochondrion. However, ATP is not produced in the matrix of the chloroplast because there is not a matrix there. Okay, so it's produced actually in the stroma. So let's look at the comparison here. If we look to the left, we have the mitochondrion structure. So remember, we have the electron transport chain. Here, the protons are going to be pumped from the matrix to the intermembrane space, building up that proton gradient, which will then drive the production of ATP because the protons can only move by the proton motive force through the ATP synthase, right? So proton motive force basically drives ATP synthase to produce ATP by combining inorganic phosphate with ADP to make ATP. And this occurs in the matrix of the mitochondria. Now let's kind of go over to the chloroplast here. So here shows the chloroplast structure. So instead of the matrix, we have the stroma. Instead of the intermembrane space, we have the thylakoid space. So what's actually happening? The protons are going to be pumped from the stroma to the thylakoid space, building up a proton gradient in the thylakoid space, which then is going to drive the production of ATP by ATP synthase. This is that chemiosmosis and that proton motive force that occurs in the chloroplast rather than the mitochondrion. So here, protons are going to be pumped through ATP synthase back to the stroma, and then in the stroma, ATP synthase is going to combine inorganic phosphate with ADP to make ATP. Okay. On the left, this is called oxidative phosphorylation. On the right, this is called photophosphorylation because the energy that ultimately drives this is coming from light. Now let's kind of get an overview of the thylakoid and the thylakoid membrane with the space and how this is actually happening in relation to the light reactions and linear electron flow. So here we have photosystem 2 and photosystem 1. Remember, light is going to be absorbed by both photosystems, where the electrons or the photons are going to be passed to the reaction center. Now, the electrons or photons that go to the reaction center in photosystem 1 ultimately are coming from the electron transport chain from photosystem 2. So remember, water enters here and gets split. Okay, so then we have those, this is where the electrons ultimately come from. And then oxygen is released. Now here, the excited electrons are going to be passed to plastoquinone through the electron transport chain to plastocyanin to photosystem 1, where the excited electron is eventually going to be passed to ferredoxin. Okay, during this process right here, this is the electron transport chain, the protons are going to be pumped actively, remember this is active transport, from the stroma to the intermembrane space, building a chemical gradient, a proton gradient, which is going to drive the production of a proton motive force. The only way the protons can go back to the stroma, because they desire to be at equilibrium at some point, the protons are going to have to go through ATP synthase, which drives the mechanical action of combining inorganic phosphate with ADP to make ATP. Now we're going to move into the Kelvin cycle in the next segment. However, adding this, remember, ferredoxin is going to accept the electrons from photosystem 1, and then it goes to NADP plus reductase, which is the enzyme that reduces NADP plus to NADPH by combining a hydrogen and electrons to it. So now we've made some ADP or NADPH. And then, of course, here, I already talked about the production of ATP, and conveniently, both of them are made in the stroma where the Kelvin cycle takes place. Again, the Kelvin cycle depends on the production of the NADPH and the ATP from the light reactions. All right, so just kind of an overview. Remember, plants kind of require sunlight, and the environment and other organisms depend on this. And the water comes from ultimately the ground. The water is going to be transported up through the vascular tissue of the plant, and water will enter the leaves. And this is where water becomes split, and then oxygen is released. Carbon dioxide is going to come in and then get fixed. The carbon is going to be fixed into sugars like sucrose or glucose, and then be transported through the plant. And then it could be transported to other organisms that might be associated with it, like often fungi are associated with the roots of the plants. Also, some bacteria are as well. Now, what we're going to do is talk about in the next segment how the carbon from the carbon dioxide gets incorporated into chemical organic molecules. This is a quick summary of this segment. I talked about the light reactions, kind of introduced them again, and of course the details of the light reactions, which include linear electron flow and cyclic electron flow. Remember, linear electron flow produces ATP and NADPH. Cyclic electron flow only produces ATP. And it involves electron transport and chemiosmosis. Remember the generation of a proton gradient and ATP synthase, combining ADP plus inorganic phosphate to make ATP.